This assay was developed to address key questions in the field of plasma membrane biology and to establish how efficiently damaged cells can reseal their plasma membrane. This technique can be used to measure the efficiency of plasma membrane resealing in a high-throughput capacity and to identify specific proteins and corresponding pathways that mediate plasma membrane resealing.
Begin by adding 20 milliliters of a 2.5 times 10 to the 5th HeLa cells per milliliter of growth medium suspension into a sterile pipette basin. And use a 10 milliliter serological pipette to thoroughly mix the cells. Next, use a multi-channel micropipette to seed 100 microliters of cells per well in triplicate in a 96-well flat, clear-bottom, black polystyrene tissue culture-treated plate.
Remember that a homogeneous cell distribution is key to a successful experiment, as comparisons between all of the experimental conditions require equivalent cell counts.
After 24 hours in a humidified cell culture incubator at 37 degrees Celsius and 5% CO2, pre-warm the plate reader to 37 degrees Celsius and set the optical configuration to monochromator, the read mode to fluorescence, and the read type to kinetic. Under the wavelength settings, select a 9 nanometer excitation and a 15 nanometer emission band pass. For assays using propydium iodide, set the excitation and emission wavelengths to 535 nanometers and 617 nanometers respectively. Under plate type, select 96 wells for the plate format and select a preset plate configuration corresponding to a black wall clear bottom plate.